Dear students, welcome to my channel, Botany Varsha. Today's lecture is for B.Sc first year, first sem students, paper second, cell biology. Topic is cell and cell wall. Topic is very familiar to all of you. It is included in plus 2 syllabus also. So, cell is the basic structural, functional and biological unit of all known organisms and is the smallest unit of life, often called the building blocks of the life. The study of cell is called cell biology or cytology. Cell was first time discovered by Robert Hooke in the year 1665 and he observed tiny compartments in a very thin slice of cork. Cell can be defined as a unit of biological activity bounded by membrane which is selectively permeable to substances and independently capable of self-reproduction in a medium free of other living system. Depending upon the presence or absence of nuclear envelope, cells are of two types, prokaryotic cell and eukaryotic cell. Prokaryotic cell in which nucleus is absent. Prominent, well-defined nucleus is absent. That means there is no nuclear membrane, nucleoplasm and nucleolus. Nuclear material is naked DNA which is scattered in cytoplasm without basic proteins called histones. Such type of DNA is called nucleode which is found in bacteria. Eukaryotic cell in which prominent, well-defined true nucleus is present. That means proper nuclear membrane, nucleoplasm, nucleolus are present and after condensation that chromatin convert into chromosomes which is made up of DNA and histones both. The eukaryotic cell includes both animal cell and plant cell. In both cells, membrane-bound organelles like cell membrane, cytoplasm, nucleus, endoplasmic reticulum, Golgi bodies, mitochondria and ribosomes are present. The typical difference between these two cells is the presence of plastid and large central vacuole which is present in the case of plant cell but absent in animal cell. But in animal cell, centrioles are present which are absent in plant cell. And the most distinguishing feature is that the plant cell has a rigid cell wall that is lacking in the animal cell. Now, this is the structure of typical eukaryotic plant cell which is composed of two parts, outer cell wall and then protoplast. Second part is protoplast. And the outermost boundary is called cell wall. It gives proper definite shape and provide protection to the protoplast. And the second part is protoplast which includes plasma membrane and all the cell organelles which covered by it. Now, I show you with the help of pointer that this outermost covering is plant cell. Outermost covering, it is followed by plasma membrane and inner portion is filled with cytoplasm. In the cytoplasm, many membrane-bound cell organelles like chloroplast and mitochondria, Golgi complex, etc. are present. Mitochondria is the powerhouse of the cell. Chloroplast is the site of photosynthesis. Golgi complex, and this is nucleus, and it controls all the metabolic activity of cell. And these are endoplasmic reticulum, rough endoplasmic reticulum, and smooth endoplasmic reticulum, and centrally located large vacuole, and some starch grains, etc. are also present over here. Now, difference between prokaryotic and eukaryotic cell. And first of all, size of the cell. Prokaryotic cell is always 1 to 5 millimicron in size, whereas eukaryotic cells are larger in size, more than 10 millimicron. And prokaryotic cells are always unicellular, and eukaryotic cells often multicellular. For example, chlorella and chlamydomonas, they are unicellular also, and most of the cells are eukaryotic cells are multicellular. And a cell wall consists of peptidoglycan, and in the case of prokaryotes, the cell wall consists of murin or peptidoglycan, whereas in the case of eukaryotes, cell wall consists of cellulose. And there's no well-defined nucleus in the case of prokaryotes, and other membrane-bound cell organelles are also absent. But in eukaryotic cell, well-defined, proper, prominent nucleus is present, and other membrane-bound cell organelles like chloroplast, mitochondria, and endoplasmic reticulum, etc. present. When we talk about the DNA, DNA is circular in the case of prokaryotes and without basic proteins, histones, called nucleoid. And in eukaryotes, DNA is linear and always found with the association of histone protein and further produce chromosomes after condensation. And ribosomes are very small, 70s, and in eukaryotic cells they are 80s. And when we talk about the cell division, it takes place by binary fission in prokaryotes, and in eukaryotes, cell division takes place by mitosis and meiosis. And reproduction is always asexual. Examples, bacteria, cyanobacteria, examples of prokaryotic cell, and examples of eukaryotic cells, plant cell and animal cell. This is a typical prokaryotic cell, typical bacterial cell, and outermost layer is a cell wall, which is followed by plasma membrane. In both the cases, cell wall and plasma membrane is present, but in few cases, in few bacteria, capsule is also present. And but main difference is the presence and absence of nucleus. In the case of bacteria, centrally located, this is circular DNA, which is without protein, and it is scattered in the cytoplasm, and there is no any nuclear membrane, nucleolus, nucleoplasm, so naked DNA is present. But in plant cell, prominent nucleus is present, and prominent outer nuclear wall, and nucleoplasm and nucleolus is also present. And but in prokaryotic cell, these pili are also present, but they are absent in eukaryotic cell. Now, the structure of cell wall. Cell wall is outermost rigid, protective and supportive, semi-transparent covering of plant, bacterial cell and fungi. It was first time discovered by Robert Hooke in the year 1665, and it is composed of cellulose, hemicellulose, pectin, proteins, water, lignin, suberin, chitin, silica and wax. And first cellulose, and it is found in the form of microfibrils, very thin, unbranched, fibrous long structures which are consist of 2000 to 25,000 cellulosic unit, and it provides strength and structure to the cell wall. Hemicellulose is also present, which is non-cellulosic polysaccharides, pectin substances. It holds water and form a gel that filled the space between the fibrous element. So it can convert into solution and gel, both the structure. Protein. Cell wall contains many types of structural proteins, and for example, extensins and expansins. Extensins provide structural support and form a barrier for the inverting microbes, also control the extensibility of the cell wall. And expansins help in the expansion of the growing cell wall. Example, alpha expansin, beta expansin, they are found in the plant cell. And a plant cell is hydrated with 60 to 75% of its mass having water. Excuse me. Lignin, it is tough glue-like substances composed of aldehyde and coniferyl alcohol units. It is cement-like material which binds and anchor the cellulose and protein components and provide rigidity, strength and hardness. Example, sclerenchymatous cells of the woody plant. Next, cutin. Cutin is fat-like substances deposited on the outer surface of cell wall as a separate layer called cuticle. It is found on the outer surface of leaf also. So outer surface, outer epidermis, outside the outer epidermis, cuticle, thin layer of cuticle is found, and the suberin. Wall of cork and endodermal cell contain special fatty substances called suberin. It is deposited on the inner side of cell wall. In some cells, tannin also deposited with suberin. Example, cork cell. And silica, it is deposited in the cell wall of some grasses, but all the substances are not found in one cell. So they are examples are different. Now the structure of cell wall. A cell wall has three parts: middle lamella, primary cell wall and secondary cell wall. First, middle lamella. Middle lamella is thin, amorphous, cementing, glue-like material which is found in the intercellular spaces between the two adjacent cells and bind them. It is composed of calcium and magnesium pectate. It is first layer which is deposited at the time of cytokinesis. During its formation, microtubules in the cytoplasm migrate and aggregate. Fibrous at equatorial plate. So small vesicles also produce from the Golgi bodies which assemble or deposited at the portion of the cell plate, which further produce middle lamella. Next is primary cell wall. Primary cell wall is found in the young cells, especially meristematic cell, photosynthetic cells, storage cells of fruits, all parenchymatous cells, and all the growing cells. Actually, it is 1 to 2 millimeter in thickness and consists of cellulosic microfibrils, hemicellulose, pectic substances, and protein. The main function of primary cell wall is it provides mechanical support and protection to the protoplast. And it is porous and permeable, so it allows transportation from inside to outside and outside to inside. Next is secondary cell wall. Secondary cell wall is found in mature cell, and it provides proper shape to the cell wall, and it is made up of many layers which are named as S1, S2, S3, and it is composed of cellulose, lignin, suberin, pectin, hemicellulose, fat and waxes, for example trachees and vessels elements. Suberin is deposited in the cord cell and endodermal cells. And some special features are also found, special structures are also found. These are small pits in the cell wall. These are the areas where secondary cell wall is not deposited on the primary cell wall, and such area is called pits. In gymnosperm, secondary cell wall partly overhang the pit called bordered pits. This is characteristic feature of gymnosperm. And plasmodesmata. Plasmodesmata is the thinner portion, and it is perforated portion of primary cell wall where secondary cell wall is not produced, called primary pit field. This area has pores from which cytoplasmic strands connect one cell to the adjacent cell. This is called plasmodesmata. Now I can show you the pictures. So you can see the picture. This is outermost layer. This is the middle lamella, which is found between the two adjacent cell, and both side of the middle lamella primary cell wall is found, and then in secondary cell wall is found, and secondary cell wall is consist of many layers, S1, S2, S3. These are the layers. And now you can see the three adjacent cell, and this yellow line is actually middle lamellae, which the main function of middle lamellae is to bind the two adjacent cells. So it is cementing glue like material getting magnesium and calcium pactate which bind the two cells. This is the structure of cell wall, which is consist of hemicellulose, and outermost portion is consist of cellulosic microfibril, then hemicellulose centrally located protein cells, and both the side pectin substances are found in the cell wall. And this is the typical plant cell, which is showing small pores or cytoplasmic strands. These are plasmodesmata. This central layer is middle lamella, and you can also see this middle lamella in the center. This white line is middle lamella, and both the side this black line, that is primary cell wall, and this white layer is secondary cell wall. So this is secondary cell wall, and outermost in the center of the two cell middle lamella is present. Now functions of the cell wall. Cell wall is rigid and it maintains the cell shape proper. It gives the proper shape to the cell, and the main function is protection because it is rigid. It is outermost covering. It will protect the inner protoplast and other cell organelles from the outer pathogens and harmful chemicals also, and it gives mechanical support to stand and keep erect the cell, and it allows transportation also, and because it is porous permeable, so it allows the transportation outside to inside, inside to outside. Many metabolic enzymatic activity are known to occur in the cell wall. So this is the end of the lecture. And thanks for watching me. Stay tuned for my next lecture. Till then, goodbye.